Hi, everybody. Just give me a couple minutes to get ready. Okay, let's first go through each of the questions, and then we'll talk about the problems like being timed out and all. Okay. Okay, question 1 and 2. Okay, question 1. Identify the part of the microscope labeled with the arrow. What's the answer?
Arm.
Arm.
Okay, arm. Okay, now keep in mind that for each one of these questions, there might be 3 or 4 more questions in a pool for question 1 that will be showing something else. So I'm going to be looking at that and making sure that everything's okay. Okay. Question 2. Identify the part of the microscope. What is that?
The ocular lens.
The ocular lens. Now, I've got to consider whether I'll take eyepiece. But I'll wait to the end to decide that. Let me write it down, though.
Dr. Benton?
Yes.
Is there a way that we can see what we scored on the test at this point in time?
No, you can't.
Okay.
Okay, here's the reason why. It's screwed up. It's one thing. But tell you what, Lily, when you come to lab or on Tuesday morning, just come over to my office and I'll put your test on and you can see how you did.
Okay, thank you.
And hopefully by then I'll have them graded. For some reason, it's not putting any grades up. The only thing that we got grades on were the 10 calculator questions. Otherwise, I've got to go through every one of these and make determinations. So, but let me write this one down. Okay, let's go down to question 3. Put my glasses back on. When putting a microscope away, what is the first step you must do?
Lower the stage all the way?
Tell me, what did y'all put down?
Completely lower
the stage. All the
Yeah,
way.
I think that's what I put also.
Off the stage.
Okay. That's correct.
Would turning off the light be acceptable?
Oh, okay. Now, this question and also the one that is what is the second thing you do? I usually tell students to turn off the light, unplug it, then drop the stage, take off any slides, rotate the objective to the lowest power of the scanning lens, and then use lens paper to clean the lenses and all. So I'm going to go ahead and write this down for questions 3 and 4. Everybody gets credit for it.
Thank you, sir.
Now, what I'm going to do is, I probably won't change it on this, but I'll just, like for these two right here and any others we have, I give you credit for it. Everybody's getting credit for it, even if you got it right. Because I got 119 of these things to grade. And I'm going to go crazy and blind. Okay, question 5. What is the total magnification when a 100x objective lens is being used?
1,000.
1,000 is correct. Anybody have any problems with that one? Good, because I'm going to keep it at that. Okay. Question 6. Okay. What is the shape of the bacteria cell Bacillus subtilis?
Oblong?
Nope.
Bacillus.
Bacillus. Bacillus, or it could be rod-shaped, or a rod. Okay.
I said bacilli because I thought that was like the singular form.
I will go through and I will check that and count it correct.
Sir, I could be just stupid, but I interpreted this as describe the shape. So something along the lines of kind of long and curved. Is that just stupidity? Or
no, it's you weren't looking close enough to the slide, because you see there's one cell here. There's a gap.
I got a Vibrio for like Vibrio.
Oh,
cholera.
You got a Vibrio. Okay,
Yes,
and what did you put for the Vibrio?
I put slightly curved with a flagellum, because that's what
Okay, well,
our reference material.
I think we're going to go to that in just a moment. Okay, here we go. What is the shape of bacterial cell Vibrio cholera?
I had this one and I put comma.
Okay, I'm going to, comma is going to be counted correct. Hold, let me write this down.
Oh, sir, I think for that question, I wasn't sure between like cocci or bacillus. I think I put cocci bacillus because it looks kind of in between that. Does that count?
No, it's not a cocoa bacilli. Just a second. Let me write down some of the other options for this one. Okay, I will accept curved, slightly curved, and Vibrio.
Sir, what if I put vibrate by accident because I didn't know how to the name?
I'll count it correct.
Sir, I think you said comma as well.
Yeah. Thank you. Okay. Now, what is the phylum of Spirillum voltans? Well, let me just say right off the bat, Spirillum is the genus. So remember I gave you all those words to look up? What is the correct word?
Proteobacteria.
Correct. Now, I'm going to go through and see if anybody got that wrong because of misspelling or a capital letter or something like that. Okay.
Dr. Benton, for this one, I put spiral.
That's the shape.
Oh.
That's the shape. I don't think that'll work.
Okay, I tried.
Okay, now, what is the phylum of the Vibrio?
Proteobacteria.
Proteobacteria also. Vibrio, again, is the genus. It's also the shape. Okay. Okay. This is a euglena. To what phylum does euglena belong to?
Euglenozoa.
Well, someone is creative here. Euglenidia. Was euglenidia one of the choices of words?
Yes.
Regrettably, it was, and that was something that, yeah,
Okay.
I don't know why that was there. It seemed to be there were two. It was either there were two options. It was like euglenozoa and then euglenida, and we had previously, like in the study guide or something, we had been given euglenophila and euglenozoa as the two options.
Yeah.
So I assumed one was in substitute for the other.
Okay, here's what I'm going to do. Everybody gets credit for it.
Woohoo!
So that's 3 of them so far you get credit for. That's 6 points so far. Merry Christmas. Okay.
And a Happy New Year.
Wait, some of these were case sensitive?
Most of them weren't. But some of them I noticed when I was looking through on some, they were case sensitive and other ones were not case sensitive. I'm going to look at it. If it was counted wrong because of case sensitivity, I'll correct it. That's why I'm going to have a long weekend. And on Monday, my eyes are going to be looking about like that. So,
Dr. Benson.
Yeah.
Would we still get the points even if we didn't get the question?
Yes, sir.
All right, cool. Thank you.
Notice, look, when I say credit for all, that means I'm not even going to look at this question when I go through it. I'm just going to skip over it. And everybody is getting 6 points or get that 2 points. Okay? Because it's easier for me, I can look and I can go through each test. If I do this, I can go through 1 test a minute while my eyes are still holding up. And so I can get it done. But if I have to go through every single one, my eyes burn out. Okay, what class does Trypanosoma belong to?
No idea how to pronounce it.
Cnidoplastiae. Look, these are all fairly recent terms. This isn't what the old books have, but that's what we're going with. And that's one of the words that was on the thing that you had to look up. Okay. To what kingdom?
Oh, yeah, I knew that one. I just still, even from the study, I had no clue how to pronounce it.
Okay. 12. To what kingdom does penicillin belong to?
Fungi.
Fungi, correct. Okay, this is Pucinium. Now, to what kingdom does Pucinia belong to?
Fungi.
Fungi. It's a fungus. Okay.
Dr. Benton?
Yes.
Oh, wait, never mind. Sorry.
Okay. Now, to what phylum does Pucinia belong to?
Basidiomycota.
Mycota. Okay. Look, if I were teaching this the way we used to teach it, I'd be teaching just fungi, and I'd go through the different phylums that we had. We have Basidiomycota because you see these things here? They're called basidiospores, and they'll be released, and each one will grow into a new one. If you have a cup right here, And inside the cup or the little opening, you have a main stalk, and then coming off the main stalk will be two little side things. And at every one of the places where it comes off, you have a spore, and that's called an ASCII spore, and that would be ASCII mycota. And then we have another group that sexual reproduction has never been observed. Doesn't mean it doesn't happen. And we call that the mixi mycota. And that'll be like the athlete's foot and that sort of thing. So, but this would be the basidiomycota, which was that one of the words you had to look up?
Yes, sir. Basidiomycota and
Okay.
ascomycota.
Okay. Now here. What phylum do Saccharomyces belong to? Y'all know what Saccharomyces is?
No, but I'm pretty sure it's ascomycota.
It's yeast. And it's ascomycota. Okay, these are yeast cells. Now, an interesting thing about yeast cells, and this is a little bit of trivia, is that for many years, they did not know it could reproduce sexually. But now, in all the years with microscopes, it has only been recorded on 4 times. And the last 2 times, it was recorded with microscopes, and one of them was an electron microscope of it, and where they found it happen. So it doesn't happen very often, but it does happen. Okay. Oh, I love this. What superphylum does Paramecium caudata belong to?
Alveolata.
Yeah, how many people got that right?
I think I did because I studied alveolata pretty thoroughly.
Okay.
But honestly, I don't remember. I've tried to block the entire experience out of my memory.
Me too.
Okay, we'll take a look at it. Okay. What superphylum does Pyridinium belong to?
Alveolata.
Yeah. Now, Pyridinium here, you see it has a dip all the way around it. And what you don't see is that there'll be a flagella down at this end right here. This is one of the ones I use when I'm teaching marine botany, because it's an ocean one. Okay. What is the defining character of Pyridinium? Now, this person here put down bioluminescence. That is a correct answer, because these, when you have massive numbers and you bother them in the still water, they'll flash. But they have cellulose plates, one here, another one there, and they also have 2 flagella, usually one long and one very short. Let's see, we'll have the short one here, and the long one will go around along this, what they call the sulcus. So, okay. But if you put bioluminescence, I'll count it right.
Dr. Benton, if we put that they were like fluorescent, I don't remember exactly what I put.
Okay, well, if you put fluorescent, I'll count it.
Okay.
Sir, we didn't all get the same questions,
No,
right?
I know. But I'm going to go through each one that y'all have had, and I'm going to do the same thing on that.
Okay.
Okay. Because there are some of them that you had, like for each one of these, you may have had 6 other questions that were randomized to everybody, but they're all very similar. Okay, but I'm going to go through those.
All right.
I think for these questions about the characteristics, I tried to list as many as I could remember. So if they are correct, would you still count it?
Yes. I'm going to check every one of them.
Okay, thank you.
I'll tell you what, let me just make it easy. I'll give everybody credit. That means you've got 8 points so far.
All right. Does that translate to 16 since this is going to be doubled?
No.
Or is it already doubled?
No. When I do everyone's just getting it. Boom. I'm going to give you the extra credit. So, Merry Christmas.
And a very happy New Year.
Oh, yeah. I didn't think anything could be worse than 2020. Okay. What phylum does Plasmodium falciparum belong to? Okay, here's where we had a problem. In your spelling, they misspelled it where there's an M. See, it's supposed to be apicomplexa, but they caught it, and so they put both here. So that one should come out okay. Now, this person gave the term anthrophyta. Anthrophyta are flowering plants. Are these flowering plants? No,
It doesn't appear so.
These are protozoa that cause malaria. Okay. Lactinophenol blue or cotton blue used on fungal cells stains which biomolecule?
Oh, it's chitin. I put peptidoglycan.
Peptidoglycan is found in bacteria, but it's chitin. Anyone want to articulate a different answer? Okay. Okay, unicellular photosynthetic organisms are the primary producers for the planet. Name the phyla of the photosynthetic organism nostoc.
Cyanobacteria?
That's right.
Dr. Benson,
yeah.
Can we go back up to question 20? If I put carbohydrate, does that count because chitin is a carbohydrate?
Let me look.
I agree with Lily. I put the same thing.
So did I.
Oh man, you made things miserable for me. Okay, carbohydrate. I'll check it. And if you put carbohydrate, I'll put it correct. Okay. Now, this is nostoc. Nostoc is a blue-green algae, and it's very, very common locally. We collect it every time I teach marine botany. We collect it and we look at it. It's a very common one. And nostoc is actually put in some types of food supplements. They do have a certain amount of vitamin B in them. Oh, and the local Indians and in Mexico, they still collect it from mats on soil that's wet all the time. And then they mash it up and they cook it like a pancake and they eat it. Okay. What is the unique feature of monophyla?
Would vascular be wrong?
Well, vascular is not unique. Okay, vascular is a very common feature.
I put that it was the first group of vascular plants, which is
that one would be correct.
Yes.
And also, see, this person wrote down comprises ferns, seedless plants with a prominent sporophyte. That's a correct answer also. So I'm going to look at those. This is 22. Okay. Okay. What is unique? What is a unique feature of Coniferophyta?
Cones.
Cones. Okay. Good. Yeah. Someone have a question?
Is that question specific? Is it just cone with an E, or will it count it wrong if it's with an S at the end?
I'll check.
Thank you.
Dr. Benson.
Yeah, just a second. Let me write this down before I forget it. Okay, yes, sir.
For number 22, would it be correct if we said the structure went from could be simple to complex to 3 to 4 times complex?
I'd have to know the entire answer. Do you remember exactly how you put it?
Something about like their design. It could either be simple to 3 to 4 times complex.
Oh. Tell you what you do. Find me a place where it's listed like that. Take a screenshot and send it to me.
Got it.
Okay.
For 23, I put cones with an S.
Okay. I just wrote down here to check for whether or not plural
Okay.
is anything or not. Okay. Okay, 24. What tissue type is indicated by the arrow? What's the arrow touching?
The epidermis.
The epidermis. So what is the tissue type?
Dermal.
Dermal. However, if you write epidermis, I'll count it correct.
For some of these questions, I don't think I had that one. I think I had the arrow was pointing to the structure below. The arrows
I'm
are
going
unclear.
to look at them, Noah. I'm going to look at them. So what do you think you had of that? A node?
Well, no. The thing is that I don't think I had that one. I think I had the one below, and I couldn't tell if the arrow was pointing to the vascular bundles or not. But I put vascular bundles.
Well, that would be
Put vascular
bundles.
tissue because I thought it was pointing to the purple like circle.
Okay.
So I put
I
vascular
thought it was
tissue.
pointing to the purple circle.
There was a different question where I put
Okay.
vascular.
If you had this one to label instead of this one, I would take vascular tissue. I would take that it's a monocot. No, my mistake. It's dicot because they're in circles right there.
Maybe
I would
take midrib.
Midrib? No, the midrib is this one right here.
Yeah, the big thing in the middle, right?
Yeah. Mm-hmm. But we're looking here.
This is a big. Oh.
This is from the stem.
Damn. Okay. I guess I just misinterpreted.
Well, hey, we do a lot of that. Okay. Now, you have, I think there's about 5-6 questions for this particular one. Identify the flower part. This one here is pointing to what?
The style.
The style. Okay, the whole thing here, female reproductive structure, is called the pistil. Okay, the pistil is made up of 3 parts. The stigma here at the surface, the style coming down, and the ovary. Okay, if you had one that pointed to this is an anther. Of this whole thing, the anther is going to be the male reproductive. Now, what do we call this one little piece right here?
Anther.
The stamen.
Isn't the stamen the entire structure and the anther is a little bit at the end?
Okay, yeah, you are correct. You are correct. Okay, so, and then what are these pink things?
Petals.
Petals. What's that green thing?
Sepal.
Okay, what's this section right in here?
Receptacle.
Receptacle. Okay, the peduncle is the one that goes farther down, but the wide part here is the receptacle. But I'm going to take a look at each of these and check them. Okay, now see here on this one, identify the flower part. And it's pointing to the steeple, but how did the person write it?
Steeple.
Steeple. So I'm going to have to give that one credit. Okay. I say, I'm going to go through and check these. Oh, this is a hard one. What phylum is indicated by this picture?
Anthrophyta.
Anthrophyta. And why did he get it wrong?
You put an R.
Okay. Okay. Okay, which phylum is indicated down below?
Manilaphyta.
Mm-hmm.
I don't see any cones.
Well, yeah. I happen to know whose paper this is, and I'll ask him personally this next Tuesday. And it's not Noah, and it's not Paul.
Oh.
So.
Wait.
So, but we were just covering this with the ferns a little bit ago. Okay, now, what class of Anthophyta does this flower belong to?
Dicot.
Dicot. Let's count the petals. 1, 2, 3, 4, 5. If the petals are in 4s or 5s, it's dicot. If they're in groups of 3, it's monocot. Oh, and look at this particular answer. I don't think that's correct.
Actually, it's correct.
Well, yeah, but not good enough for the points.
Wait, so it's not my paper. Unless it is, and I just blanked out.
No, it's not your paper.
Okay, cool.
You'll find out on Tuesday.
Okay.
If the person lets me announce that it's theirs. I don't think he will. Okay. Now this one. What shape does this flower represent? And actually, it's not one flower, it's a floret. Okay, tubular, cruciform, rotate, or lingulate. Take a look. What does it do? Okay. Rotating would be correct, but then the most correct one is, you see these little tubes coming off to each one? That would make it tubular. But if you have rotate, I'll give it credit also.
Sir, I'm not sure, but I never saw any of those terms or that photo specifically.
Okay. That means you got one of the other ones. I'm going to be checking these each. Okay.
No, like in the labs, like in the slides they give us.
Yeah, well, I think this was in the photos on the online section. But tell you what, on Tuesday's lab, bring this to my attention again, and I'll look over it to see what we have and on what we covered. And if you are correct where we didn't do anything on it, then I'll give everybody credit for it.
Cool, thank you.
Okay, I just need to go back and check everything to make sure that that's the way it is. Okay. Name a shared feature of the Coniferophyta and the Bryophyta.
That they're both land plants work?
Well, some of your Bryophyta are actually, they live in bogs.
Personally, I've never seen a bog, sir. I don't know where you got that from.
I know. We don't have bogs here. You got to go up to the northern areas of the U.S., or you go to England or France and Germany where they have the bogs. So
Was that one of the labs, sir, that we missed due to the virus? A trip to some European area to observe?
Oh, yes,
this
yes.
particular
Oh, it was a
item?
fantastic trip. I'm sorry you couldn't make it.
Yes, I'm sure.
Okay, this is question number 33. Okay, I'm going to go through and check it. Now, if half of the people missed it, I'll give everybody credit for it. Has to be half of them miss it.
Well, I don't mind saying I did my part.
Yeah, okay, good. Okay.
Dr. Benson.
Yeah.
For that question, would you accept they're both autotrophs and use photosynthesis?
Well, that is a shared feature.
Uh-huh.
So I guess that would be it. That would be counted correct.
All right, thank you.
No problem. See, that's why I have some of these like this, I have to go through and make a determination on each one. But I'm being very generous on interpretations right now because I hate this damn test to no end. Okay, now we have a leaf coming up. To what class does it belong to? Monocot or dicot?
Mono.
Why?
Parallel venation.
Parallel.
You got it.
Parallel.
Parallel venation. So it's,
I have a question.
yeah.
Some of the answers I might have put like an S after the end, like monocots or dicots. Will it still count it right?
I'll look and see if it does answer the question. If it does answer the question, or it could be counted right. I mean, you know, the way we discussed it and everything, I'll count it right.
Okay, thank you.
Okay. See, on things like this, on monocot, this is a fairly simple one, but when we get to some others where it's not very clear, like we did on this last one up here, besides both are terrestrial or they both do photosynthesis, you know, but you see the chlorophyll there? Okay. That's why I'm going, one of the reasons I'm going through each and every test to make sure that everything is correct. Okay. Now, what type of pattern is represented in this leaf?
Palmate?
Nope. Palmate is where you have one
Pinnate.
thing and then it each goes out like that, like a palm tree. This is what? Well, oh yeah, palmate, but the vein pattern. Okay, palmate will be correct, but the most the best one is net venation. But here they're calling it palmate. Palmate is more like what you'll find in a palm, but I'll count it correct.
Would you accept branched or branching as an answer to this one? As opposed to net?
Let me consider it when I'm going through it.
Thank you, sir.
No problem. Because even on monocots, it branches from one original place. So, but then they're parallel once they've branched off. And that is usually at the base of the petiole or right here. Okay. Okay. Taking a look at this stem, to which class is it representing? Monocot or dicot?
Dicot.
Dicot. Why?
The vascular bundles
Location
are
of the vascular bundles.
in a circle.
Right. Okay. Now, some of you all may have gotten one where the circles were in the middle or kind of spread around. What would that be?
Monocot.
Monocot.
Monocot. Okay. Okay. Identify the part of the root with the arrow. Okay. Anyone want to articulate an answer?
Ground tissue.
I said meristem.
Okay.
It's very difficult to see where the arrow is actually pointing to.
Yeah, I know. It's going to this tissue right here, which would be ground tissue. But you don't know exactly, is it going towards the vascular or, and see here, lengthening. Okay. This part does lengthen. Not necessarily this part up here, but this part is because, see, this part is where the meristem is, right here. And so it's making new cells. When you make new cells, are they as large as the adult cells? Nope. So this section in here is where the cells have been created, but they're small. So as they grow, would there be lengthening? A certain amount. So I would count lengthening on that. What are some of the other answers y'all wrote?
I didn't have this one.
Okay.
I put ground tissue.
Okay. I'll be taking a look and seeing what all other people put. And if it's reasonable, I'll count it correct. Okay. If population A has a Shannon diversity index, that was in the lab. Guess what? I'm giving everybody credit for that one. Because I didn't cover it. Okay, everyone gets credit for it. How many is that? 4? 8 points? Or is that 5?
I think that's 5.
5, 10 points. Okay. Well, we still got 4 more questions. Okay. Is a p-value of 0.49 significant?
Not even close.
Okay, what does it have to be significant?
Less than 0.05.
Right. You are correct. But
was there a question on the exam that had it at 0.049?
There
because
was.
I would have swore that's the question I had, and the answer then is yes.
Okay.
Yep.
Yeah, because it is less than 0.05.
Right, just barely.
Oh, okay, I'll take a look at it, Noah.
I also got that question.
Okay, and yeah, if it's 0.049, it is significant.
Yeah, I also had that question.
Okay, I'll go through and check that on everybody. That'll just be one of them I go through. Okay. A non-significant p-value on the t-test indicates what?
That the null hypothesis is correct?
Yeah, that there is no difference. And the null hypothesis is correct. Okay.
For that one, if we specifically said that there's no difference between the means of the two groups, would that be correct?
All you have to do is say that it shows that there's no correlation. But okay. I'm going to read these answers and see what you wrote, because there's a lot of different ways it can be written. Oh boy, okay, that was, what was that? That was question 38. Okay. Let's go down to the next one. Name one of the two hypotheses that explains high rates of autogamy.
Reproductive assurance.
Okay. Was this from your reading material?
Yes.
Okay.
Yes, sir.
Yes, it was. The last two questions were from the scientific paper.
So, did anyone put something different than these? Okay. Question 40. The study examines the relationship between the breeding system and the three floral traits. Name one of these traits.
Anther
Okay.
stigma distance.
Anther to stigma distance, pollen to ovule ratio, flower size. These are all the different traits. Did anyone else put another trait down? Nope. Okay. So right now, everybody automatically gets 10 points. And then I have to go through and check all these other questions. Now, the way the test is made is where you get 1 point per question. So I'm going to come up with a total before I add these. I'm going to double it on my spreadsheet, and then I'll add the 10 points, and then I'll go to your test, and I'll pop in all the grades there. Okay, I hope to have that by Monday. Okay, now let's go to the second part of the test. By the way, did anyone get kicked out so much that they could never complete this thing?
I had at least one question that I missed due to the system having an issue, but that was the worst it was for me.
Okay. Well,
Oh, I don't know if you guys talked about this already because I joined like a while ago, but when I was like clicking submit, it said that my answers weren't saving, and then it said that I didn't answer the questions, but I did. So I don't know if my answers are there.
Hold on a minute. That's one I have to check in.
That happened to me too.
Okay. First of all, who was the first person that was speaking?
Me, Josephine.
Okay. See, I don't see it coming out on. I don't have everybody's pictures up. So okay, it's Josephine.
Yes.
Okay, and what section are you in?
I'm in the online section, like the completely online.
Yeah, I know. All of my labs have both online and present.
Oh, okay.
Okay, are you in FM, DM, DDM, or EEM?
I'm in EEM.
EEM, okay. I will personally check that. Okay, who else was there? Melissa?
Yes, and I'm section EEM.
Okay, you're also EEM?
Yes, and I also sent you an email about it, but I mean, I kind of just told you right now.
Okay, when did you send the email?
I sent it right after I took the test.
Ah, okay. Yeah. I will personally check both of yours to make sure that you have it. And if you don't, then I'll send you emails.
Okay.
Okay. And can you also check the email too, though? Because I added something else in there. Like I said something else.
Okay. Hold on just a second. Well, what day did you take the test? Monday, Tuesday, or Wednesday?
Wednesday.
I'm trying to figure out the when you sent the email.
Wednesday.
Okay, just give me a moment here. Okay, hold on just a second. I can't get to my email. Oh, wait, hold on. Okay.
Yeah, I wrote a lot. I kind of wrote down the answers of what I put in
Okay,
each, like
can
the
you
ones
see it
that
on the
didn't let me.
screen?
Yeah, I can see it.
Chihuahua. Okay, hold on. See, normally it doesn't follow along. Okay. Okay. Okay, I'll be going through each one of your questions and checking them. Okay. And I see that you took it twice.
Yeah, because I thought it would have it was
don't
just, like,
worry
the first
about
time,
it.
so I tried it again.
I gave you the opportunity to do so, and I'll take the higher grade. But I'm going to go through and check it. Okay.
Okay, thank you.
Okay.
Oh.
Yeah.
I have one more thing. I wasn't here in the beginning, so I'm not sure if this was discussed already, but when I was like going through the respondus, it didn't take me through the proper like steps. So because usually it says like if your camera's working, but it didn't turn on at all. It just
I
says
know.
the test,
I know.
So I don't think I have the
It
video.
didn't for anybody. Okay.
Oh, okay.
It didn't for anybody. You all saw how many headaches I had, and I had to keep rechanging the options because they kept switching back to a default overnight, sometimes after 15 minutes. Okay. Let me go to the just a second. Okay, let me go to the stats test. I'll put it on the screen in just a minute. Okay. Okay, can you see the first question there?
Yes, sir.
Yes, sir.
Okay. What is the field of view in millimeters for this 4X lens? Okay, so let's see here. There's one full one, two full ones, three full ones. And it looks like this is the right on the edge. There's that before. So it would be just under 4.5. So, but I will check it to see if you're within the correct, if you're in the ballpark. Okay. What is the field of view in microns at a 40x objective if the field of view is is on the 10 power and it's 1.6 microns?
Dr. Benton, not about this question, but would you check the questions like question 1? Because I think for my field of view question, I was not shown bars, or it was in no way communicated that there was a unit of measurement that could be used.
Okay.
Yes, sir. Mine looked like just a normal bacterial slide sample.
Yeah.
Mine as well.
We've got some of those. I'll check each and every one of them. Okay. So this will be the calculus. And that'll be, yeah. See, there's only 10 questions in this, so I can go through them fairly quickly and make changes. And I'll check each and every one of them. Okay. Okay. Question 3. Convert the value of 145.1 microns to millimeters. What do? You move the decimal point. Okay? And you're going to move it this other way. So, hold on, let me see here. Yeah. Okay. Let's go to the next one. Number 4. Convert the value of 0.456 millimeters to microns. It'd be 456 microns. Okay. Now, we're starting into I think when you're using the
Terrace diversity?
Yeah. Okay, calculate the values for the final column of the table for species A to 3 decimal places. Okay. Did anyone have problems with that? So everyone was good with it?
Yep.
Okay.
For me.
Huh?
I just said it was fine for me.
Okay. Okay. What is the Shannon diversity index in a given population to 3 decimal points? Well, remember, I didn't cover Shannon diversity index, so everyone gets credit for question 6. So that takes you up to, what is it, 12 points?
Yes, sir. Thank you.
Okay. Question 7. What is the median for the given data to 3 decimals?
Dr. Benton?
Yes.
So when I would open up the Excel files, the copy that it opened in, I don't know what was happening, if it was like a problem on my end, but I was not able to like open the descriptive statistics program.
Okay. Who's this that's talking?
This is Riley Hale.
It's what?
Riley Hale.
Okay, hold on just a moment. And what section are you in?
I am in, I believe it's DDM. I was able to do some of it.
Okay.
But I ended up having like 45 seconds.
No, you're not the only one. We had a bunch of them, even in the on-campus ones, that got kicked out because the modem was too slow. Not the modem, but the Wi-Fi was too slow, or Blackboard was too slow. And some of them got kicked out because it said it's overloaded and try again later. So I'm going to give you credit for those.
Okay, thank you.
Okay. And
Dr. Benton, do you also want me to give you my name? I was on campus.
Yeah.
Angelica Longoria.
Okay. And what section?
DM.
DM. Okay, anybody else? Okay. Angelica, when you come to lab on Tuesday, we'll together go through your test. We'll look at it and go through it.
Thank you, sir.
No problem. Okay, question 8. What is the sample variance of the data to 3 decimals? Okay. Should be 0.314. Anyone have problems with that? Oh, did anyone have a problem? Okay, let me put it this way. You had to use the Excel stats for data. Is there anyone that couldn't access that?
I just did all mine by hand.
Oh, okay. Did you come out okay on it?
I think I got that one right. Eventually I worked it out, but I had like 30 seconds to do 3 questions, so I don't know how accurate.
Oh man. Okay. 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 9. Using the given data, perform a paired t-test. Now this one I know we had problems with because some people couldn't get the stats to work. Is there anyone here that had problems with that? Because we had some people that they would highlight and it wouldn't stay selected and all. If you had problems with it, let me know during lab or whenever you can get to these things and take a look at them. Okay. Number 10, using the given data, perform a paired t-test. What is the one-tail p-value? Anyone have problems with that?
No, sir, none at all.
Okay. Anybody else? Because not everyone is up on it as you are, Noah. We're all not as perfect as you.
Oh, well, yeah, just keep on believing that. Keep on believing that, and don't look at my exam score.
Okay, so right now, everybody, oh, and by the way, I'm going to add these 10 questions to the other 40. Okay, and you're going to get a number. I'm going to take that number and I'm going to multiply it times 2. And then I'll be adding 12 points to it. Some of you will actually be getting more because I'm going to check on these other things and I'm going to give you credit if you got certain answers that are reasonable. Okay? Any other questions? No? Okay. Well, I'm going to start on these and I'm hoping by Sunday night I'll have it all on my Excel and then I'll take the scores and I'll put them on Blackboard. Okay?
So for the points,
Thank you so much, sir. This has been a lifesaver.
Okay, Noah. Someone else was trying to speak. Who was it?
Oh, it was me. So about the points, so you add the extra points to both this exam and the 40 question one. So you're combining them and then doing all that, right?
Yeah. Okay.
Oh, okay.
You had 40 questions on the fill in the blank. You had 10 questions on the calculator one. That's a total of 50 questions. And they're going to be 2 points each. So, but the way the test is designed, you only get 1 point. So I'm going to take those points, multiply it times 2, and then I'm going to add 12 points to it.
Okay, thank you, Dr. Benton.
Okay, look, this is like last semester. But, you know, some of you all were about ready to commit suicide after the midterm or after the final. But when we went through it, and then I went through each and every person's test, did it change your grades?
Significantly.
Yeah, it made them better.
Significant. Yeah. There was a significant difference. The p-value was very low. So I'm planning on doing the same thing here. And within the next week or so, all of your labs are going to be graded and put on my spreadsheet, and then I'll start putting averages down on Blackboard. Okay?
I have a question. So someone told me from like one of the other classes that we could like turn in extra credit stuff.
Okay.
Like
you have a diversity project, and I'm not doing the diversity project that they're doing on that's on the lab. Okay, I'm going to be adding to your materials, course materials page, in which it's going to have community service extra points. Okay, if you do some type of community service, for example, we have a number of students going down to do beach cleanup. We've got other ones that are helping elderly neighbors mow their lawn and do things like that. I've got one student that is volunteering at a hospice. I've got another one that spends a lot of time with the old people at the nursing homes. You find some type of service thing that you're going to help somebody or you're going to clean up the environment. You take some pictures of it, of you doing it, and then get a note written by somebody that of what you were doing and signed. And you remember how you last year or last semester, you submitted your lab journals to me?
Yeah.
In other words, you took pictures of it and you sent it to me.
Uh-huh.
That's what you'll do here. And if you do that, see, I'm being very lenient on the type of service you can do, because used to be, I could say, we're all going down to do beach cleanup at such and such a date. And I'd get a bus from MSA and take everybody down. But we can't do that because of COVID. So you're having to find things at around your home or things that you can do. And you have until the week before the finals to find some type of service, take pictures of it, get someone to sign a little note stating what you were doing, and then you either scan it or photograph it and send it to me. Okay? And that's 15 points that will be added to one of your either the midterm or the final lab practicum. Seeing how it's probably going to be the week before the end, I'll probably add it to the final one. Okay.
Okay. Thank you.
No problem. It's my pleasure, and you all, I don't really have to say much about doing service. A lot of you are already doing volunteer work and servicing. So I'm very proud of you all for that. And I respect you greatly for taking that into your own account and doing things. So, okay, any other questions? Yes.
I'm in a nonprofit organization and I get hours by creating posts, like Instagram posts, and my friend like gives me the hours.
Okay.
So what about the picture? Do I just send you a picture of the Instagram post?
Have someone take some pictures of you doing the Instagram post, and then have your friend write a little letter and sign it of what you were doing. Okay?
Okay.
Because there's nothing wrong with that, particularly since it's a nonprofit. Now, if you're getting paid, then that's something different. But you're a volunteer.
Yeah, I'm not getting paid. It's a nonprofit.
Okay. Well, in some nonprofits, people get paid. But no, you're volunteering, so that's completely okay. Okay.
Thank you.
And I'm very, very proud of you for doing that. So, okay. Any other questions? Okay. Well, I'm going to shut it down and I'm going to start working on these things. So.
Thank you, Dr. Benton.
No problem.
Thank you so much, sir. Have a great one.
Oh, no.
Thank you, sir. Thank you. Have a good evening.
Thank you, Dr.
Thank
Benton.
you, sir.
Look, you all are worth it. Okay? You all are worth it. So, okay. By those of you that are in the Edinburgh group, get there a little early on Monday because we're going to start the skinning of the cats. Those in the Brownsville groups, I'll have the labs open by 12:30 if you want to come in and start skinning the cats. Okay? Okay. Oh, I've asked MSA for some gloves, but if they don't get them, can you all bring gloves with you?
I
Yes,
can.
sir.
Okay, fantastic. I'll try to bring some from here too. Okay, I appreciate it. Thank you all very much.
Sir, what time would be best for Monday?
Oh, for Edinburgh,
Yes.
it'll be open before or around 1 o'clock. Oh, and
Okay.
by the way, Edinburgh, last week I forgot to take you the pizza. So I'll have the pizza out in the hallway or in the other lab and not where we have the cats.
Alrighty. Thank you, sir.
And I highly recommend you eat the pizza before you handle the cats. Because for some reason, even when you wear gloves, it seems like the smell leaks through to your fingers. From experience. Okay. Anything else?
For the gloves, is it latex gloves?
It can be latex, it can be nitrile. My own personal preference is the nitrile. Okay, but it can be either. But do not get the ones that they give for food handlers in cafeterias. You know, those clear polyethylene ones? Mm-hmm. They leak like a sieve. Oh, they're bad. So if you have either latex or you have nitrile, they're okay. Or if you want, okay. Do you have dishwashing gloves at home?
I think so.
Those work pretty good. But I wouldn't suggest you take them home and use them in the sink after you use them on cats.
No.
But they work great because they have the little ridges that will help you hold on to things. So, okay, that'll be it then. And I'll see you all, some on Monday, the rest of you on Tuesday and Wednesday.
All right, I'll see you on Monday, sir.
Okay, have
Bye.
a great one.
You too.
Dr. Benson.
Yes, sir.
Is the biodiversity project still on?
Yeah.
Because I had sent you an email regarding the bibliography section.
Oh, no. Listen. I'm doing a different diversity than what the Edinburgh people are. And this is where you do some type of service project.
Oh, okay. That's what you meant. But I thought that was just an extra, like a separate part.
No. And I'm going to give everybody that does it, something like that, 15 points on probably the final lab practicum.
Oh, okay. Thank you, sir.
Oh, no, my pleasure.
All right. Have a good day, sir.
Okay, you too.
Hey, Dr. Benson.
Yes.
Whenever I finish taking my test, I don't know if it's like the system or if it's just normal. I can't see the answers that I put. So is that something to worry about?
I'll check on it.
Okay.
Okay. What, Sophia? What section are you in?
This is Anaya. I'm in DDM.
Oh, Anaya? Okay.
Yes.
Hold on. Okay, DDM?
Yes.
I'll check it.
Okay, thank you.
Okay, no, my pleasure. My pleasure.
Have
If there's anything wrong, I'll email you.
Okay.
Okay?
Have a good day.
I'll take care.